Hi, my name is Avery Seward, and I'm a senior at Hopkins majoring in behavioral biology and minoring in psychology. For the last year, I've been involved in a research project in the Riddle Laboratory in the Department of Orthopedic Surgery. Today, I will be presenting some of our most recent work on how PPARg gamma in osteoblasts regulate skeletal homeostasis and body composition. The closely related conditions of type 2 diabetes and obesity result in impairments in metabolic health that include insulin resistance, hyperglycemia, and impaired fat metabolism. Many organ systems can be impacted by these disruptions in normal energy homeostasis, and they can lead to reduced quality of life as a result of other linked health problems, including cardiovascular disease, nerve damage, osteopenia, cancer, and even mortality. Since the prevalence of these conditions have now reached pandemic levels, they represent a public health challenge and have economic costs of more than $100 billion. The focus of the work I will share today is the interaction of metabolic disease and the skeletal system. Obesity and diabetes can lead to reductions in bone strength, leading to an increased risk of fracture. Bone can also contribute to the regulation of metabolism through the production of endocrine factors. Our laboratory has focused on the nuclear receptor PPARg gamma and a bone-derived protein called sclerostin, which both play critical roles in regulating metabolism and have become a focus for therapies aimed at improving insulin resistance. The PPARg gamma nuclear receptor plays a role in adipose tissue development and the regulation of glucose metabolism through its actions in a number of tissues, including fat, muscle, and liver. These functions made PPARg gamma a target for type 2 diabetes and obesity treatments. Current PPARg gamma treatments like thiazolidinediones work by activating the PPARg gamma receptor and have been shown to improve insulin sensitivity. However, these therapies are unfortunately also linked to cardiovascular disease, weight gain, and bone loss, which limits their use clinically. A protein called sclerostin encoded by the SOST gene is crucial for bone growth and remodeling. The evidence of sclerostin's role is shown in conditions of Van Buchem disease, in which sclerostin is impaired. You see bone overgrowth as seen in the photos on the right. Further, outside of bone, clinical studies have identified a correlation between serum sclerostin levels and fat mass in diabetics, as well as healthy adults. Previous studies from our laboratory showed that in addition to regulating bone mass, sclerostin also regulated fat mass and glucose homeostasis. Previous sclerostin knockout mice showed increased bone volume, reduced fat pad weights, and improved insulin sensitivity, all which are shown in the figures below. We are integrating what we know about PPARg gamma and sclerostin to test our hypothesis that genetic inhibition of PPARg gamma will influence body composition and glucose homeostasis through its regulation of sclerostin expression. In our laboratory's research, we used an osteoblast osteocyte-specific PPARg gamma knockout mouse model to investigate the role of PPARg gamma signaling in osteoblasts and the effect it has on metabolic parameters, bone parameters, and the overall phenotype of mice. The figure on the right just confirms our model worked, showing that PPARg gamma mRNA levels are significantly reduced in the PPARg gamma knockouts. PPARg gamma mice show an increased trabecular bone volume and a larger cortical tissue cross-sectional area. Looking at mice fed a regular chow diet, our results show that PPARg gamma knockouts develop a decrease in subcutaneous fat mass. These mice also show reduced expression of genes involved with fatty acid synthesis, as shown on the left, and an increased expression of genes involved with fatty acid oxidation, as shown on the right. Further, PPARg gamma knockout mice had improved glucose metabolism, as shown on their glucose and insulin tolerance tests. This means that PPARg gamma knockouts remodeled their adipose tissue in an effective way to prevent weight gain and control aspects of their metabolism related to type 2 diabetes and obesity. Next, we fed mice a high-fat diet to challenge their metabolism as in conditions of type 2 diabetes and obesity. We found that PPARg knockout mice maintained a lower body weight and reduced fat mass in many tissues of the body when fed a high-fat diet, including gonadal and inguinal white adipose tissue, brown adipose tissue, and the liver. This means that these mice were protected from weight gain, which we see in the wild-type mice, and suggests that the PPARg knockout mice had a more effective metabolism. The PPARg knockouts displayed an increased glucose tolerance and insulin sensitivity when on a high-fat diet. Overall, this means that the PPARg receptor can adapt under conditions of metabolic stresses seen in type 2 diabetes and obesity. Further, these mice had a significant decrease in their expression of the SOS gene and a corresponding decrease in serum sclerostin levels. Our ongoing studies are designed to try to raise the sclerostin levels and see if it returns the metabolic phenotype to normal, meaning more similar to wild-type mice. Overall, our data indicated that PPARg in osteoblasts contributed to the regulation of whole body metabolism. PPARg in osteoblasts likely does this through its regulation of sclerostin expression. Our research has value in bettering type 2 diabetes and obesity treatments and other metabolic disruptions, as well as could help protect from bone loss and related conditions such as osteopenia and osteoporosis. Additionally, better understanding the link between the skeleton and the rest of the body is valuable and could help further our understanding of both orthopedic and endocrine disorders. Our next steps would be further understanding sclerostin's role by looking at if a clinical pharmacological agonist of PPARg that is known to improve insulin sensitivity regulates sclerostin expression and how sclerostin is involved in efficacy of the therapy. We wonder whether a dual therapy involving sclerostin could improve the effectiveness of a PPARg agonist while also protecting from bone loss. These are my references, and I would also like to thank my mentor, Dr. Ryan Riddle, for allowing me to learn and be involved in his laboratory's work. Thank you so much for taking the time to listen to my presentation.